Whether you're a bioprocess engineer hunting for the perfect scaled-down model, a biotech founder architecting the leap to commercialization, or a scientific analyst who craves raw technical truth, you've reached the technical edge. In this space, we live by a single mantra: the process is the product. Welcome to the Biomanufacturing and Fermentation Technology podcast with Prasad Ernala. Hit subscribe and ring the bell to join a global community and stay at the vanguard of biotech translation. Let's build the bioindustrial future together.
Today's part, one of 4 parts of QbD with the theme of discussion is From Philosophy to Biological Reality. This part explores the application of quality by design, QbD, principles to microbial fermentation, moving away from traditional reactive testing toward a proactive, science-based development framework. It highlights how the inherent variability of living systems, driven by genetic drift, nonlinear metabolic shifts, and environmental interactions, requires a more sophisticated approach than simple one-factor-at-a-time experimentation. By utilizing ICH regulatory guidelines, manufacturers can link a product's clinical intent to critical quality attributes and specific process parameters. The discussion emphasizes using structured risk management tools, such as FMEA, to identify how upstream biological fluctuations propagate through the manufacturing lifecycle. Ultimately, the material frames QbD as a disciplined strategy for navigating biological complexity to ensure consistent product safety and efficacy.
Welcome back to the deep dive. Today we are tackling a topic that, I'll be honest, has a bit of a branding problem. We're talking about quality by design, or QbD. And I can already feel some of you tensing up just hearing that.
It doesn't exactly scream thriller, does it?
It really doesn't. You know, when most people in the industry hear QbD, I think they instinctively reach for a coffee or check their watches.
Yeah, that's it. They think of a massive regulatory checklist, endless templates for the FDA.
Just a mountain of bureaucracy standing between them and actually getting a batch out the door. It feels like homework.
That's the reputation it's earned, unfortunately.
Yeah.
It's often viewed as a compliance taxa hoop you have to jump through so you can get back to the real work.
But, you know, if we look at our source material for this deep dive, Foundations of Quality by Design in Microbial Fermentation, a very, very different picture emerges. The authors argue this isn't about filling out forms at all.
No.
They present QbD as a survival guide.
That is the perfect way to frame it. It is absolutely a survival guide for managing the inherent chaos of living systems.
And that's our mission today. We aren't building widgets here. We're not stamping out metal parts where the steel behaves the same way every Tuesday.
Right.
We are wrangling self-replicating cells that, they change their minds, they mutate, they get stressed out, and sometimes just decide to die on us.
So we want to move from that abstract philosophy of quality
down to the biological reality. How do you tame that messiness with a structured, risk-based approach?
And that shift from quality as a paperwork exercise to quality as a tool for biological control. That's the heart of what we're discussing.
So let's start with the history. The source material draws a pretty sharp line between the old way and the new way. They describe the traditional approach as almost forensic.
Like an autopsy, yes. Historically, the industry operated on what you'd call a test quality in model.
What does that mean exactly?
Well, you ran your process, and you just sort of accepted that manufacturing variability was a fact of life. You hoped for the best.
And then?
Then, at the very end, you tested the final product against the Pharmacopoeial specifications.
So you bake the cake, and only after it's out of the oven do you check if it's poisonous.
Effectively. And think about the economic cost of that. If the test fails at the end, the batch is enjoyed. It's already lost. You've wasted weeks of time, expensive materials.
It's purely reactive.
Purely. You launch an investigation, maybe you file like HAPI, a corrective and preventive action, but you're looking in the rearview mirror. You aren't controlling the car. You're just measuring the skid marks.
The whole mindset was that the process is a black box. Stuff happens in the tank, we don't really get it, so let's just check what comes out.
Precisely. The new way, this science-based design, it completely rejects the black box. The core shift here is treating variability not as a mystery, but as a controllable outcome of a process you actually understand.
So the goal isn't to test for quality at the end.
No, it's to design a process where, if you stay within certain parameters, you know you are reliably delivering quality.
You design it.
You're designing it into the process itself.
Okay, now this is where we run into that alphabet soup of regulations. The source mentions the ICH framework, and I feel like we have to briefly demystify this. We've got Q8, Q9, Q10.
Q11, Q12. It sounds like a droid roll call from the sci-fi movie.
It does.
But they actually tell a pretty cohesive story if you look at them as a narrative instead of just a list.
So walk us through that story. What's the structure?
It starts with ICH Q8. That's pharmaceutical development. This is where the whole concept of a design space lives, proving you understand the link between your inputs and your outputs.
Yeah.
Then you have Q9, which is quality risk management. That's the brain of the operation, how you assess and control risk.
Yeah.
Q10 is a quality system that wraps around all of it.
But the source says it gets really interesting for people in manufacturing with Q11 and Q12.
Yes. Q11 is specifically for the drug substance.
The actual goo in the fermenter.
Correct. And then Q12, I mean, that is the game changer. That's lifecycle management.
That's the one that caught my eye. Because it implies the regulators don't just want a snapshot.
No, they want a movie. They want a framework where your process understanding evolves over time. You don't just file it and forget it.
You update your control strategy as you learn more.
Exactly. It acknowledges that you will know more about your product in year 5 than you did in year 1.
Which brings us right to the biology. The source material identifies a very clear villain in this story, and it's not the regulators.
No, it's biological variability.
The central constraint of the whole industry.
Absolutely.
The text makes a really interesting comparison between a chemical reactor and a fermenter. If I'm running a chemical process, my catalyst is, I don't know, platinum. It sits there, it does its job.
It doesn't have a bad day.
Right.
In a chemical process, your reactants are defined, your stoichiometry is defined. The variability comes from physics mixing, heat transfer.
But in fermentation,
Your catalyst is the cell population. And it is self-replicating. It's adaptive. It is literally evolving while you are trying to use it to make a product.
it's like if your spark plugs decided to change shape halfway through a road trip.
Or maybe start reproducing in the engine block.
Mm-hmm.
The source outlines 3 specific layers of chaos here.
Okay.
The first is nonlinearity and path dependence.
Unpack that for us. What does that mean?
It means small differences at the start can snowball. In a robust chemical system, if you nudge it, it often settles back. In biology, if you nudge it, say, your inoculum quality is slightly lower, or the lag phase takes an hour longer.
It might just go off in a completely new direction.
A totally new direction,
Right.
because the cells are changing state. A longer lag phase might mean they consume a different nutrient, which triggers a different metabolic pathway. And suddenly your whole process trajectory is different. You have to look at the history of the batch.
And then there's the second layer, genetic and metabolic drift. This is the stuff that keeps engineers up at night.
This is literally survival of the fittest happening in your tank. These production strains are under immense metabolic burden. We're forcing them to make a drug they don't want to make.
So they look for a way out.
Of course they do. They can mutate. They could lose the plasmids that carry the instructions for your drug. Or they might just shift priorities. Switch from growth mode to survival mode.
And that's where you see things like proteases pop up.
Exactly. The enzymes that chew up proteins. So now, not only are you not making product, your cells are actively destroying what you've already made.
And the third layer is what really blew my mind. Interaction-driven variability. The source says variables don't work in isolation.
This is critical. In a simple system, you think, if I increase the feed rate, I get more growth. Simple. But in fermentation, the effect of the feed rate depends on the dissolved oxygen, which depends on the pH, which depends on the temperature. It is a web.
A web that gets even more complicated when you scale up. The scale-up trap.
Yes, scale-dependent heterogeneity. Imagine a huge 20,000-liter tank. You can't mix that perfectly. So you have gradients, zones with high oxygen near the impeller, zones with low oxygen near the walls.
So a single cell floating around is basically going on a roller coaster ride.
That's a perfect analogy. It travels into a nutrient-rich zone, turns on its growth genes, then it gets swept into a nutrient-poor zone, gets stressed, turns on a starvation response. It's constantly cycling between these states.
And a small-scale model would never see that.
Never. A small beaker is easy to mix,
Yeah.
but in the big tank, that oscillation can completely change the product quality.
Which explains why the old one factor at a time method of FAT is such a
Well,
disaster.
it's so dangerous in biology.
But it feels so intuitive, right? Yeah. Change one variable, keep everything else constant, see what happens.
It feels intuitive, but it lies to you. OFAT assumes factors act independently. It assumes if you find the best temperature, and then you find the best pH, you can just combine them for the best result.
But because of that web of interactions,
That assumption just falls apart. You might find a local optimum, a little peak that looks good, but you completely miss the global optimum because you never tested how temperature and pH interact. OFAP blinds you to the true landscape of the process.
Okay, so if biology is messy and simple experiments don't work, how do we actually design quality? The source talks about this logic chain from QTPP to CQA.
This is the structure QbD provides. It starts with the QTPP, the Quality Target Product Profile.
The voice of the patient.
Exactly. It's what the drug needs to be. Strength, purity, stability, safety. That's your starting line.
And from there we get the CQAs, critical quality attributes.
Right, and this is the bridge between that clinical need and your manufacturing process. And this is where it gets tricky because the source highlights a trap here. The difference between what's convenient to measure and what actually matters.
The streetlight effect. Looking for your keys where the light is, not where you drop them.
Precisely. In bioprocessing, we love measuring things that are easy. A simple UV purity test gives you a nice number. But does it tell you if the glycosylation pattern on your protein has changed?
And glycosylation matters because...
Because the sugar structures on a protein can determine how long it stays in the blood, or whether the patient's immune system attacks it. That simple purity test might miss a change that makes the drug totally ineffective.
So QbD demands that you measure the hard stuff if the hard stuff is what matters to the patient.
Yes. Even if the analytical development is a nightmare, you have to link the attribute to the clinical performance.
There was another trap mentioned that I think is really relevant, the titer versus quality trap. Everyone wants high titers, more product, more money.
It's the classic vanity metric. But high yield does not equal success. The source gives a great example. If you push the cells too hard for high titer, you just force feed them, you might saturate their folding machinery.
The assembly line gets jammed.
Exactly. The helper proteins that fold your drug just can't keep up. The proteins come out misfolded, they form aggregates. So you might have a tank full of protein, but half of it is garbage that you now have to filter out.
So you have to look at yield, product quality, and impurities all at the same time?
You have to. You're optimizing for the effective yield of safe product.
Okay, so we have our attributes. We know what to look for. Now we have to talk about risk. The source brings up ICH-Q9 and risk thinking.
And this is just formalizing what good scientists do intuitively. ICH-Q9 defines risk simply. It's the probability of harm times the severity of harm.
And the tool they mention is FMEA, failure mode, and effects analysis. How does that apply to a fermenter?
Well, it's not just listing broken parts. You have to link the equipment to the biology.
Yeah.
And FMEA here isn't just what if the pump breaks. It's what is the biological consequence if the pump breaks.
So the pump stopped becomes...
The pump stopped, so the cells starved for 20 minutes. And the timing matters. A feed pump failure early in the growth phase has a totally different impact than one during the production phase. A pause early on might just delay things, but later...
Later on, that starvation event could trigger that stress response we talked about.
Right. Or cause the cells to start eating the product they just made. FMEA forces you to map those specific biological consequences.
This leads to the concept of the failure chain. I found this fascinating. It's like a domino effect inside the tank.
This is where you really see the interconnectedness. Let's take the scenario from the source. Say you have a low inoculum, just a little bit less than usual.
Okay, so you start with fewer active cells.
Which leads to a longer lag phase. The cells take longer to get going, but maybe your feed pump is on a timer. So the feed starts at hour 10, but the cells aren't ready.
So now you're dumping food on cells that aren't eating, nutrient excess.
Which triggers overflow metabolism. The cells start producing things like lactate or acetate. That lowers the pH. The controller adds base to fix it, which increases the saltiness of the broth.
And suddenly the environment is toxic.
And that stress alters the impurity profile. So a low inoculum didn't just mean less product. It meant different, dirtier product. That is the failure chain.
And this chain doesn't stop at the fermenter. The source talks about upstream impacts downstream.
No, and this is a huge friction point in bioprocessing. If you mess up the fermentation, say you have high cell lysis because the run went too long, you are dumping all those host cell proteins and DNA into the broth.
And the downstream team has to deal with that.
And if you haven't designed for that risk, you overload their chromatography columns. You ruin expensive resin. You let impurities slip through. If you don't control the upstream risk, you are just passing the buck downstream.
So when we put all this together, the philosophy shift, the biological chaos, the logic chain, the risk management, what's the big takeaway?
The synthesis is that QbD is a disciplined method to confront complexity. It takes us away from just tweaking knobs until the number goes up. It forces us to map what the patient needs, all the way back to the process variables.
It transforms bioprocessing from just following a recipe to managing a risk strategy.
Exactly. Every parameter, every control limit, is actually a risk reduction measure. You aren't just setting pH at 7.0, you are maintaining pH at 7.0 to prevent protease activation. The intent is totally different.
It gives purpose to the parameters.
It does. And it changes how you troubleshoot. You stop looking for the broken part and you start looking for the broken interaction.
I love that. Now, before we wrap, we always like to leave our listeners with a final provocative thought. What does this deep dive suggest that maybe isn't immediately obvious?
I think the most provocative idea hidden in here is that the risk hypothesis is actually a living construct.
What do you mean by that?
Well, we tend to think of quality as a fixed target. The drug is the drug, the specs are locked. But the source implies that as we learn more about the biology, our definition of what is critical might actually change.
So the CQA isn't set in stone.
It shouldn't be. Maybe next year we discover that a tiny impurity we thought was harmless actually causes a side effect. Or we develop a new assay that sees something we couldn't before. Suddenly, a non-critical attribute becomes a critical quality attribute.
Which means the process design is never truly finished.
Exactly. It's constantly refined as our biological knowledge grows. We're chasing a target that becomes clearer the closer we get to it.
That is both terrifying and exciting. The work is never done, but the product is always getting safer.
Precisely. It keeps us humble in the face of the biology.
A great place to leave it. We hope this deep dive helped you see the method behind the madness of fermentation. Thanks for listening.
See you next time.
Hi there, thank you for staying with me until the very end. If these technical insights help bridge the gap in your own process, please hit subscribe and make sure to hit the notification bell. Stay connected for our next deep dive into the realities of biomanufacturing. I am Prasad Ernala. Thanks for listening.